My name is Urvashi Tiwari, and I'm presenting my topic on evaluation of selected indigenous spices and herbs derived small molecules as potential inhibitors of SIRAP1, means straw regulatory element binding protein 1, and its implication for breast cancer using MD simulations and MMPBSA calculation, means molecular mechanics, Boltzmann, Poisson, Boltzmann surface area. Now, let's start with the introduction. It is widely recognized that breast cancer often increase the lipid increment for the proliferation and development of cancer cells. And this lipid is synthesis is required by strong elementary regulatory binding protein 1, and dysregulation in SIRAP1 may increase lipid production, which reprogram the cancer cells and its proliferation. So that's why I'm choosing SIRAP1. The question arises why I'm choosing the protein SIRAP1 as a target, because SIRAP1, first condition is SIRAP1 is linked with the metastasis and proliferation, and because it increased the lipid metabolism when is dysregulated, then then it increases the lipid metabolism, which is the first step in the cancer cells. While the SIRAP1 is divided into two parts, SIRAP1A and 1C. SIRAP1A is responsible for all SIRAP genes, all SIRAP responsive genes, and SIRAP1C is responsible for lipid metabolism. The difference only between the SIRAP1A and SIRAP1C isomer is their transcriptional start point, which is different from each other, and that's why I'm choosing this as a target protein. To target this, I selected indigenous spices and herbs as an inhibitor. Why? Because it exhibits strong anti-cancer and anti-mutagenic effects, and they can modify, suppress, and regulate the cells. And last but not the least, these are cost effective and with less or minimal side effects. That's why I selected indigenous spices, which we use in our daily life for food. So these are 20 most common Indian spices and herbs that we use in our daily life. It includes turmeric, whether it is cardamom, black cardamom, cloves, cinnamon, black pepper, cumin, and 20 compounds these are. And this is the flow chart of methodology which I done. This is my work chart which I done to calculate and evaluate the compounds. So here, the indigenous spices, and on the basis of review papers, I selected 53 anti-cancerous compounds, and then I screened it on the basis of Lipinski rule and ADMET, and evaluated 82 compounds which we use to dock with SIRAP1 chain. And by the help of two molecular docking softwares. First one is AutoDock and second is DocThor. So firstly, I have done DocThor, which is free online server present. And then after it, to validate the DocThor results, I also do the manual docking condition, that is AutoDock. And by this, I evaluated dioxane, smilezanin. I also take one control compound, means reference compound, to compare these two compounds, that is betulin, which is from Betulinaceae family and the only known inhibitor which is inhibit the SIRAP1 chain. So for this evaluated compounds, I have also checked the stability for another method. For this, I use molecular docking simulation process in 100 nanosecond, and on the basis of different parameters and MMPBSA calculations, I choose dioxane, smilezanin, and then after it, by the help of PC analysis and Sci450 metabolism biological activity spectrum, I selected dioxane, smilezanin because there is not much difference between the calculated values after these all calculations. I choose dioxane, smilezanin for my further studies. Now, the Lipinski rule, which I use to filter out 253 compounds, and I get 197 compounds which I again used admit to evaluate, and 82 compounds were eliminated. 82 compounds were used, and for admit result, and then I found 82 compounds total. So basically, Lipinski rule and admit I have done to check the oral bioavailability of my compounds and their efficacy and safety process for the drug development. And I find the 82 compounds are most potential for the docking. These are the top filtered compounds. These are the main compounds found in these spices and herbs, which I got from the filtration or screening. And after this, I do molecular docking. Molecular docking, the interaction between the ligand and protein structure. So for this, Doctor and Autodoc I use. And firstly, I use Doctor because it is online server. To validate this, I use Autodoc. And after this molecular docking, which is basically used to interact the ligand protein on the basis of their binding energies. So for this, I use this, and these are the diazenin and smileyzenin and betulin. These show the best result. And manual docking, we find that minus 6.57 binding energy was for diazenin, and for Doctor it is minus 8.27. So that's why I found that diazenin is the most plausible hit against the syrup, while the smileyzenin and the control compound betulin is a little bit low from the diazenin. That's why at this point I evaluate that diazenin is the best, but because smileyzenin is not in a much difference, that's why I also took smileyzenin for further studies. So after that, at last I found that diazenin and smileyzenin as a premium molecule for the efficient interaction with syrup 1A chain. So these are the results which I calculated and evaluated from the molecular docking. And these are the 3D and the 2D structures. This A figure shows the 3D structure in which the green condition, or the green condition is green color is defined by betulin, which interact with the protein. And then this is a B figure, shows the interaction of ligand and protein, and this is the 2D structure. So here we can see there is a amino acid which is present in the outer area of the figure. These are the amino acids which are present in the syrup protein binding site, and these colors variation shows the variations in the interaction process of the ligand and protein interactions. And this green color shows the conventional hydrogen bonding, which means to anchor the ligand into the binding site of the protein. And then after it, this alkyl condition basically shows the alkyl group of ligand which binds to the binding site of the binding region of the syrup. And then trialkyl, which shows that the ligand interaction in the system with the alkyl efficacy of the protein. So these are the 2D structure which shows the binding efficiency and molecular docking of ligand protein with ligand and protein interaction complexes. And this is a result of diazenin. Here there is one more thing, which is van der Waal interaction. Van der Waal interaction also shows the efficacy of ligand and protein binding. And then after it, conventional hydrogen bonding, carbon hydrogen bonding, alkyl, and then next one is pi alkyl. And these the corner in the below the structures, I also mentioned the energies, which is minus 8.27 result. This is result of diazenin binding energy. And then after it, this is the result of smileyzenin with energy minus 8.29. So on the comparison of these molecular studies, I also found that there is a little bit difference between the smileyzenin and diazenin, but these are comparatively higher than the betulin. So in this case, I found that smileyzenin diazenin is good potentially. That's why I used it for the further evaluation. So for this, to check the stability process, I used the MD simulation process, and MD simulation I used to evaluate by the help of GROMAS package. and in the duration, sustainable duration of 100 nanosecond. So in this, we check the stability and the behavior of the complex. So this is a result of RMSD and RMSF. RMSD, root mean square deviation, and root mean square fluctuation. The first picture is predict the root mean square deviation. So here we can see that there is colors graphical presentation, and this is the colors. Magenta color is shown by betulin, and then orange color shows by diogenin, and the violet color is sourced by smileygenin. So in this condition, we can check, also calculate the RMSD value too, which shows that smileygenin is little bit better than diogenin and betulin. And the value is for betulin is 1.907, and for diogenin is 1.926, but for smileygenin it is 1.733. So in this condition, we can say that it is not much deviate in comparison to Diogenes betulin, because the RMSD basically shows the deviation of the complexes when the ligand bind with the protein. Okay, so next graphical representation is of root mean square fluctuation. So it's shows how the residues fluctuate when protein interact with the ligand. Okay, so this fluctuation and the peaks of the fluctuation shows the flexibility and the fluctuation of residues when ligand bind with the protein. So, okay, sorry, the bell is ringing.
1 minute to finish.
Okay, I got it.
Oh, you still had some time.
Okay, sir.
That's 30 seconds.
Sorry for the delay.
So please answer on the questions in the chat.
In the chat.
Okay,
yeah.
Okay, sir.
Yes, and thank you for a very interesting presentation. Thank you.
Okay, there is a question based on your figure number 3. Your ligand is outside the active site. Okay, sir. There is a problem of the active site is the molecular document, because there is no active site is currently available for the CERV1. Betulin is also used to inhibit the CERV1, but not directly. So
Okay, stop.
Thank you.
Talking, but answer in
Thank you so much, sir. Thank you all for listening.